So one of the things I've alluded to before is this quandary of if you've got a universal tree, the three-domain tree I showed you before, how do you determine the root of that tree? In order to root a tree, you have to have an outgroup. That's the function of an outgroup in any phylogenetic analysis. And it's impossible, of course, to have an outgroup in a universal tree, by definition. Except that you have to remember that the trees you're generating are not trees of organisms. They are trees of gene sequences. It is possible to have an outgroup for a universal, for a tree containing homologous genes from all organisms, because many genes in all kinds of living things are the result of gene duplication and specialization. And so, for example, EFTU, elongation factor TU, which you, I'm sure, remember from biochemistry or some such class. It's a protein involved in helping the ribosome process from one codon to the next. This protein is homologous to EFG, another protein in the process of translation elongation. In fact, the two associate with each other. These two proteins, EFTU and EFG, are homologous to each other. And they are present, homologs of them are present in all kinds of living things. So what if you make a single alignment that contains EFG and EFTU sequences from different kinds of creatures? You will create a single tree, a universal tree, of both proteins together. And that tree will look something like this. It will be splayed out like this if you looked at them as a dendrogram. In this case, then, the EFG sequences can serve as the outgroup for the EFTU part of the tree, and vice versa. And so if you focus then on just the EFG part, here's a tree of EFGs. EFTU is the outgroup, and here's the root of that tree. And the same goes in reciprocal for EFTU. And another way to look at that is to collapse these in terms of organisms. And so here you have G and TU in the same organism, and here and here. And it's absolutely clear that the root of this tree is a short distance on the branch toward bacteria, grouping archaea and eukaryotes on one hand and bacteria on the other. Now that's a big claim. And so you've got to have corroborating evidence for that. But EFTU and EFG aren't the only proteins or genes that were duplicated earlier than the last common ancestor. Other examples are some ATPase subunits, alpha and beta. My favorite is elongator and initiator tRNA methionine. So organisms have two methional tRNAs. One to start translation, and one to put a methionine in the middle of a protein. And they're homologous to each other. Another one are some aminoacyl tRNA synthetases. The leucyl and isoleucyl tRNA synthetase are quite similar. They're very specific, and all organisms have each. But they're clearly homologs. But that split that created the separate synthetases occurred before the last uncommon ancestor. And you can do this trick with any of these, and you get the same result. And so what that means is that here's another way of looking at that three-domain tree. You've got eukaryotes, archaea, and bacteria, and the root is here. And this is exactly not what people thought they would get. The presumption would be the old eukaryote-prokaryote division, right? That is that the root would be here instead and have the archaea and bacteria on one hand and the eukaryotes on the other, but that is not the case. Archaea are genealogically more closely related to eukaryotes than they are to bacteria. Yeah?
How did they know that these gene pairs split before the last common ancestor?
So they are obviously within each one. So the question is, how do you know that they're ancient duplications? All organisms have them, and they are homologous to each other. And so the only other way that could happen is if the genes are being transferred around. But because if you do a phylogenetic tree with either of these independently, you get a perfectly reasonable looking tree, which would discount the notion of horizontal transfer. So there are two ways to think about the archaea. This doesn't have a big impact on how people think of bacteria, but it does have a big impact for how people think about these kind of creatures. If you're going to divide all living things into two kinds, you've got bacteria and everything else. In a real way of thinking about it, and this is very useful for certain circumstances, archaea are a primitive early branch in the eukaryotic tree. Wouldn't it be nice? I mean, eukaryotes are complicated, right? They've got genes and introns and all kinds of structure, and I tell people all the time, eukaryotes are too complicated. If you want to study the fundamental aspects of eukaryotes, it would be really nice to have a eukaryote with a small genome and simple cell structure, something primitive that tells you where all these eukaryotic complexities came from. And those organisms are archaea. You can learn a lot about how eukaryotes work by studying archaea. And this actually has been the major impetus for people studying archaea as a primitive early branch in the eukaryotic domain. And so, for example, yeah.
So, in like middle school and high school, we learned basic biology, biochemistry. We were using models from whole body.
Yeah.
Would we be able to get more accurate biochemical information from studying archaea about humans?
Right. So that depends. If you're looking at, for example, I don't know, DNA polymerase, it doesn't matter what organism you're studying. DNA polymerase works the same in all living things, so E. coli is convenient. But what if you're studying RNA polymerase? Bacteria have a single RNA polymerase, right? And a variety of sigma factors that bind to that and modulate their binding to different kinds of promoters under different circumstances. Eukaryotes have 3 RNA polymerases with specialized function. RNA polymerase 1 transcribes ribosomal RNAs, RNA polymerase 2 transcribes messenger RNAs and some other small RNAs, and RNA polymerase 3 transcribes tRNAs. How do you understand the evolution of those 3 RNA polymerases? You study them in these things, because archaea have 1 RNA polymerase. And it's a dead ringer for RNA polymerase 2. And it binds to a tau binding protein. And the promoters are tau boxes, just like RNA polymerase 2 in eukaryotes. Ribosomal RNAs and transfer RNAs are transcribed in archaea by that same RNA polymerase with a different transcription factor. So it's a great way of studying how those things work. RNA methylation and pseudouridylation in archaea works just exactly like in eukaryotes and not at all like bacteria. And so it's a way of studying how these weird complexities in eukaryotes originated and what the fundamental characteristics are. Does that make sense?